So today I'm going to be talking about DNA transcription. And DNA transcription is basically the process by which an RNA sequence is produced from a DNA template. So first step is when the RNA polymerase binds to the DNA. And this is when specific nucleotide sequences tell RNA polymerase where to begin and where to end. And the RNA polymerase attaches to the DNA at a specific area called the promoter region. And here the RNA polymerase separates DNA strands and synthesizes a complementary RNA strand. And the four components are adenine, cytosine, guanine, and uracil, because in RNA, thymine is replaced by uracil. And they're the covalently bonding ribonucleoside triphosphate that align opposite their exposed complementary partner. So adenine and uracil are matched together, and guanine and cytosine are matched together. Oh, and the bonding happens by using the energy from the cleavage of the additional phosphate groups to join them together. Oh, and from this picture, it shows how the RNA polymerase separates the DNA strand and how the RNA synthesizes a complementary copy right here, and then the double helix will form again. The second step is elongation, and in this step, the transcription factors unwind the DNA strand and allow RNA polymerase to transcribe a single strand of DNA into a single-stranded RNA, which then becomes mRNA, and that stands for messenger RNA. And once the RNA sequence has been synthesized, the RNA polymerase will detach from the DNA molecule and the double helix will reform. And then a sequence of DNA transcribed into RNA is known as a gene. And the last step is termination. And here the RNA polymerase moves along the DNA until it reaches a terminator sequence. The RNA polymerase then releases the mRNA polymer and detaches from the DNA. And because transcription occurs in the nucleus where the DNA is, so once the mRNA is synthesized, it moves into the cytoplasm for translation to occur. And that is DNA transcription.
All right.